Hi there, and welcome to this video on GCSE Biology for the AQA specification focusing on cell structure and in particular eukaryotes and prokaryotes. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started. Hello, and welcome to tutorial 1 of 6 on cell structure. So today we'll be looking at eukaryotes and prokaryotes, and in subsequent tutorials we'll be delving further into the world of cells. So these are the key learning objectives of this tutorial. Perhaps use this as a rough framework for your revision notes. So we'll first be looking at the structure and properties of eukaryotic cells, then compare that to that of prokaryotic cells, and finally we'll be looking at this topic in terms of size and scale. So just to give you a little more of a conceptual look on this topic, just take a moment to think of yourself right now. You may think you're made up of just one organism, yourself, but if you just take a closer look, you'll realise that inside your digestive tract you actually have around 100 trillion bacterial cells living there. This means that your body actually consists of the two major types of cells, prokaryotes and eukaryotes. So these are the main specification points we'll be covering in this tutorial, so please feel free to pause the video at this point just to read through the points because I'm going to skip ahead here. So first off, let's take a look into the structure of eukaryotic cells. So eukaryotes are organisms whose cells have a nucleus enclosed within membranes. So remember that the nucleus, which we can see over here, houses the cell's genetic material. Eukaryotic cells include plant and animal cells. Let's just take a look at the cell on this slide. It has a nucleus contained within the cytoplasm, which is surrounded by a cell membrane. Also found in the cytoplasm are many mitochondria and ribosomes. And we can draw multiple comparisons between these cells, something that we'll be exploring in subsequent tutorials. Now let's just contrast the animal cell we saw on the last slide with the plant cell, which we can see on this side. So again, we can see our nucleus, our cell membrane, our cytoplasm, our mitochondria and our ribosomes. But we can spot three differences here between the animal cell we saw on the last slide and the plant cell on this side. First off, we have a cell wall which encloses the cell around the cell membrane. We also have chloroplasts. And we have the structure in the middle, the vacuole. So now that we have an idea of what eukaryotic cells are, let's talk about prokaryotic cells. You may want to pause the video at this point to read the specification point. A prokaryote is a simple, single-celled organism that lacks a nucleus and membrane-bound organelles. We'll talk more about the nucleus and organelles in the next tutorial, but what I want you to keep in mind for now is that prokaryotic cells are not divided up on the inside by membrane walls, but consist instead of a single open space. So bacteria are classified as prokaryotic cells. And just a little interesting point here, pro means before and kary means nucleus. So around 3.5 billion years ago, long before the first plants, people or other animals appeared, prokaryotes were the first life forms on Earth. And hence, the fact that they've got pro and kary in their name kind of gives you a hint about this. Prokaryotic cells are much smaller than eukaryotic cells and also much more simplistic. So let's draw more comparisons between the two. The structure of a bacterial cell is distinctive from a eukaryotic cell because of features such as an outer cell wall. Also, bacterial cells do not have a nucleus. Instead, they have this loop of circular DNA that you can see over here. Overall, bacterial cells have a general lack of membrane-bound organelles. Prokaryotes are unicellular, i.e. they're made up of one cell only. One prokaryotic cell forms a prokaryotic organism. In contrast, eukaryotes can also be multicellular, so that means made up of more than one cell.
eukaryotes also tend to have a true membrane-bound nucleus. So let's summarise the key differences. So cell theory states that all living things consist of cells which are either prokaryotic or eukaryotic. So first off, size-wise, prokaryotic cells, that is to say bacteria, are amongst the simplest of organisms. They are made up of simple single cells with a cell structure that is simpler than the cells of eukaryotes and much smaller. Most are 0.1 micrometer to 5 micrometers in diameter. This contrasts to a eukaryotic cell, which can range between 10 and 100 micrometers in diameter. Exam-wise, note that you don't need to memorise these values. Just be aware that prokaryotic cells tend to be much smaller than eukaryotic cells. Comparison point number 2 is that eukaryotic cells contain membrane-bound organelles, whereas prokaryotic cells don't contain membrane-bound organelles. Furthermore, eukaryotic cells contain a membrane-bound nucleus, whereas in the prokaryote we have free-floating chromosomes. Also, in the eukaryotic cell we do not find plasmids, whereas in the prokaryotic cell you can see these circular loop structures of DNA here, which we refer to as plasmids. Also, ribosomes are found in both eukaryotic and prokaryotic cells, but ribosomes are much smaller in prokaryotic cells, which kind of makes sense if you think about it, given that prokaryotic cells are generally much smaller than eukaryotes. So crucial for you to remember here is that both eukaryotic and prokaryotic cells have cell membranes. Some eukaryotic cells, such as plant cells, possess cell walls, whereas all prokaryotes possess a peptidoglycan cell wall. In addition, prokaryotes tend to possess capsules. So the capsule enables the cell to attach to surfaces in its environment, but don't worry too much about this for now, we'll be exploring this a bit later on. And I mentioned a little earlier the little wiggly thing attached to the prokaryote here, it's called the flagellum, and this is a motility structure. And now we come on to our final specification point of the session. So you need to be able to make order of magnitude calculations in addition to using standard form. So differences in size are often described as differences in order of magnitude. That's the difference calculated in factors of 10. Just remember that we always talk in context of metres. So for example, the stem centi means the same as moving the decimal place 2 places to the left. So if I just draw that over here for you. So centi is equivalent to writing something like this. And as you can see, we've moved the decimal place 2 places to the left and added in a naught in that space. There. Milli is the same as moving the decimal place 3 places to the left. So, for example, I'll write that here. So that's our milli, and let's just contrast that to our centi. So can you see the pattern? With centi, we've moved the decimal places 2 places to the left. With milli, we've done 3. 2, 3. And in the same way, micro is equivalent to 6 places to the left and nano as 9 places to the left. So again, if I write those, we've got our micro here and nano would be like this. And if I just recap for you, we have just moved the decimal place 9 places to the left. So these prefixes describe progressively smaller and smaller measurements. And standard form is used when working with very large or very small numbers because you wouldn't want to be writing out all those zeros in general practice. So to summarise, in this tutorial we have covered what defines a eukaryotic cell and how this differs to prokaryotic cells. We finished off by getting to grips with size and scale terminology. In the next session we shall explore the structure and function of prokaryotic and eukaryotic cells further. But for now, well done on getting through this tutorial, and if there are any parts you are still unsure of, remember you can rewind this video or cross-check against the corresponding chapters.
Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.